Okay, welcome to our New York State Integrated Pest Management Academic Seminar Series, designed to increase awareness of new research and techniques that advance IPM and its adoption in all types of management settings. These seminars are once a month, so please check our website, which I just linked to in the chat, for our upcoming seminars and other events like our What's Bugging You First Friday series. And now it's my great pleasure to introduce Dr. Clarence Swanton, Professor Emeritus from University of Guelph, where he's had an excellent career, if I say so myself, on weed ecology and the development of integrated weed management systems for field and horticultural crops. Dr. Swanton obtained a bachelor's in botany from the University of Toronto, a master's in agrometeorology from the University of Guelph, and a PhD in plant ecology from University of Western Ontario. He's an elected fellow of the Canadian Society of Agronomy, Weed Science Society of America, and the Canadian Weed Science Society, where he also served as president. So thank you, Clarence. Take it away.
Well, thank you. It's unstable. Okay, hopefully you can hear me okay?
Yes, thank you. Yep.
Okay, so you let me know if there's any problem. Thank you very much, Brian. It is a real pleasure for me to be a participant in this series and a delight to share some of our results. So I'm going to talk today about really about how weeds affect the crop survival. And before I begin, I want to acknowledge on this slide that there are several members of the research team over the years that have contributed to these ideas and some of the data that you're about to see. I could have titled this differently. It's also relevant to why early season weed control is important and how weed seedlings emerging with the crop cause yield loss. So it's really about, I'm going to focus on the crop in this case, not on the weed. And I'm going to try to change or add to your understanding of competition. Okay? I'm going to try to present some new material. Some of it hasn't been published yet. The majority of it has. But try to change how you view the mechanisms, because there is this idea that we understand competition, and certainly we think of competition in terms of resources, and no one's going to argue that one. But I think there's more to it. I think there's a lot more than that we don't know about competition. And I'm going to share some of the physiology work that we've been doing, both at the cellular level and then at the whole plant level, to try to convince you about some novel mechanisms. And I have 4 mechanisms I'm going to talk about today that play a key role. So just to begin, I wanted to, these ideas sort of started to gel many years ago. When we were doing competition studies. And one of the things that we learned very quickly was that weeds that emerged with the crop are by far the most competitive. And that these weed seedlings, if they're not removed shortly after emergence, you can see yield losses, okay? And then came the question, well, if it's resources, how can that be, right? And this is an example of a slide that I use. You can clearly see there's no competition for light, okay? And I would argue there's no competition for nutrients because we just put fertilizer in the ground for that corn crop, so there should be lots of fertilizer. And generally, we plant to moisture, there's lots of moisture in the ground, and you can see there's lots of space. So the question is, well, if it's not resources, what else could it be? And this would play out in the work that we did around critical periods. We did a lot of work initially trying to define when you really do need to control the weeds in terms of yield. And so we did a lot of work on the critical period. And I just want to draw your attention to this weedy graph here, that part there. And one of the things we noticed in the variations of these graphs that you can generate is that very early on, you can see that this slope has a negative slope to it, and that the longer the weeds are there, truly, the greater the yield loss. But the question is, in this area up here, how can we be having a yield loss and when the crop is at the seedling stage and the weeds are at the seedling stage as well? So part of this basic premise that I want to introduce is space. But I think there are primary mechanisms that actually occur prior to any of those resource-dependent variables coming to play. And I'd like to suggest to you that it really begins with plant communication. How plants detect one another, how they detect neighbors. And it has been a fascinating area to look at how these plants interact and what happens to their physiology when they interact. So we're going to spend some time today talking about, okay, so we get the far red and red light coming in and red deficient light coming in, and it hits the weed species. And the weed species, it reflects off the weed species. And the corn crop or the crop can pick this up through the phytochrome that's in the leaves. So that protein will pick up these signals. This will allow the plant to detect its environment, okay? And we also believe that it's capable of detecting whether there are some siblings nearby or whether there are strange competitors nearby. And so that's really what we're going to focus on. And we're going to focus on that light reflection. And I'm going to show you an example of how we do that in labs. So this is some recent work we were doing on Arabidopsis, but it shows the treatments. So on the far left of the screen here, you can see that this represents our weed-free control. And the red to far red ratio is 1.4. We see grass species around the outside of that, and there is no contact between the roots. There is no shading in there, but there is this transfer of light signals, and the light signals can be picked up by that Arabidopsis seedling. And here we're approximately 0.3 in the red to far red ratio. And then we actually have an artificial, like the light, a filter or a light combination that gives us a signal identical to the biological system. So those are the three types of treatments that we use. So the first mechanism that I want to talk about is that as soon as a plant, a seedling, detects a competitor, a weed species, we pick up an enhanced production of singlet oxygen. Okay. Now, singlet oxygen is beneficial in the cell. It is useful in terms of cell signaling. It plays a key role in photosynthesis. But when it's in excess levels, it can be associated with significant cell damage. And that damage can be caused when a plant is under stress. So I'm going to give you some illustrations of what that looks like. So to understand what you're looking at right now, it's a soybean leaf. The bright spots on in the screen represent singlet oxygen. We can stain for that, and we can catch the flash of the singlet oxygen here. And so this is kind of, in parallel to looking at, perhaps you've seen maps of North America from the satellites at night, where you see New York City, just a great big white light of New York City, and then you see the countryside get darker around it to the next city. This is very similar to that. So the more bright lights there you see, the more singlet oxygen. So if you look at the weed free, that's sort of the normal background. But if you swing your eyes to the right and you see that leaf glowing in singlet oxygen, there is significant damage here. To put it in Star Trek terms, perhaps, you might think about the ship is on fire because you have excess singlet oxygen, you have damage occurring, and someone or something has to put that out in order to survive. Because if you don't put it out, you die. And so here's Arabidopsis that we've been recently working on. And you see the same thing, the control on the left. We'll call the biological, think of that as the weedy one. You see it's bright and shiny, and also under the artificial light, you see the same response. And we also see that in corn. And in corn here, this is the center stem in corn, in a corn leaf, and you see these bright leaves. So we know that this is occurring on a wide range of crop species upon early exposure to to weeds. So we're looking at early exposure of a few days, and you can get these types of signals happening in a seedling. So on your left here, this is just something we recently published in Frontiers. We have our control plant here. This is Arabidopsis. Here's our biological. We sampled a number of plants, 25 individual plants. So this is a subsample of it, just to showing you the range in the singlet oxygen explosion that happens just with the presence of a weed species, okay? And if we found out how to measure that recently. And so if you look at mean inflorescence here, the black is weed-free. Our weedy system, you can see how much more inflorescence there is there. And then under artificial light, again, it's slightly higher. So the point being that exposure to far red light, whether from an artificial source or from an alternative plant, can cause this explosion in singlet oxygen. And that creates a problem. Now, I'm going to show you, some of you may have seen this before. I've used this slide, these series of slides here to illustrate how significant this singlet oxygen can be. Now, what you're about to see for the very first time is how one plant can kill another plant, okay? And I want you to watch this plant down here on the bottom right lower end in particular, okay? Now, most plants will find mechanisms to put out the fire, okay? That is, they're going to reduce that singlet oxygen level so that they continue to survive. But under conditions of stress, it may not be possible and such that the concentration of the singlet oxygen surpasses a threshold, and that threshold is beyond the ability of the plant to survive, and it will die. So I want to show you here, this is a tobacco plant. And on the left, you have the weed-free environment. And on the right, you have it surrounded by a grass species and sending as far red light. And we treat these plants the same. In this case, we treated it with a compound to slow down electron transfer in the mitochondria. So it causes the electrons to back up. And that creates a stress in the plant, and you get an explosion of singlet oxygen. So we do that both to the control on the left and onto the right, but the plants with the weeds also have the weed species. So I want you to watch now what can happen, how one plant can kill another. And in this case, it's the ring of weeds that's going to cause damage to the tobacco plant. So this is at 0. This is at 6 hours after you start to see the damage occurring on the plants there. This is 12 hours. This is for 24 and 48 hours. And excuse me, that's 6 days later. Okay, let me walk through that again. I blew my timing there. Sorry about that. This is 6 or 12 now. I've forgotten 6, I think. 12, 24, and then 48. There we go. And you can see the plant on the right is dead. Okay, and there's 6 days. And so what happened there, the plant was not able to reduce the level. And you can see the plant on the left, even though it had the same chemical treatment, was able to do that. So that's a situation where just to show the potential damage from singlet oxygen, you got to pay attention. If you're a plant, you've got to pay attention, and you have to expend energy in order to control that. And this energy flow between dealing with these stress features are an important part of why I think plants lose yield so early. And here's an example. The plant on the right is a soybean plant, and we did the same with soybean. Okay, and you can see the plant on the right is basically well on its way to death. In this case, we were playing with the singlet oxygen genes, and there's 1,931 responsive genes in Arabidopsis plants that will respond to singlet oxygen. 57% of those, or excuse me, 57 of those responded to far red treatments. And of the 57, only 6 were common to both the weedy treatment and to the far red treatment. So we think there's a unique mechanism by which plant-on-plant interaction, it triggers certain genes to help deal with this particular problem. This can also happen in the field. We did a short little field test to show that this can happen under field conditions. And in this case, what we did, we applied what's called ALA, and ALA basically slows down the process of chlorophyll production in a plant. Okay. And so we treated, under weed-free conditions, we treated the grass species here. And you can see that grass species is pretty dead 48 hours after treatment with that compound. It's right there. And 3 weeks later, you can see that it has grown back successfully. It resurrected life after death. And so there it is, it has survived. But you take that same situation there and you treat it when there are other weeds around it, and there's the plant that goes down again. And when we came back, it has disappeared completely, okay? So it died out of that population. So in a sense, it's like self-thinning. You know, there's a, how do plant communities self-thin? And that may very well play an important role in self-thinning. So again, this is not where the plant wants to be. It wants to be able to reduce that, that death. But in order to do so, it has to expend a fair bit of energy, and it has to draw energy from other resources, other ways, other mechanisms. And that's important for you to realize what's happening. I show you this for one particular message. And so this is Arabidopsis. We had it exposed to the weed species and treated with the stress treatments again. And you can see under the weedy condition, the red and far red, you can see the plants are starting to die. And also under the, you can see damage occurring on the Arabidopsis under 72 hours. You can see that it is starting to show damage to the cells. But what's interesting here in this slide is if you had the weeds present and you control the weeds in the crop, so the weeds are going to cause an explosion of singlet oxygen, and you control the weeds in the crop, there is a legacy impact of that singlet oxygen explosion. And so here in this graph, I can't seem to find my marker to show you, but if you look under the 72-hour one, under the 72 hours, so 72 hours, we had removed the weed species, i.e. you controlled the weeds. So in a practical scenario, you think, okay, we've got the weeds controlled. There's no more damage happening out here. But 72 hours after we controlled the weeds, the plants continue to die back. And you can see how they change from the slide before to this slide. If you look particularly at the column there of 72 hours, the top one being the control, and the middle one being the weedy species, and that bottom one being the artificial system. So there's a legacy impact. Even though you took the weeds out of there, that plant has to still deal with all the chemistry and all the biochemistry because there's still issues. The cell remains on fire, and I have yet to control that. So the plant is surviving, but you can see that it was significantly impacted by that damage. So that's important to know, because you might feel pretty good about getting the sprays out there on the crop, but that damage, there continues to be a legacy impact. It also plays a role in the field around cover crops, how we manage cover crops, and how we do it, plant. So, a lot of interest in planting green. And I think planting green can be a dangerous way to handle a cover crop based on our work. I think the cover crop should be dead by the time the weed seedlings emerge. And I'll show you why I say that. Because I want to show you some data with soybeans at the hypocaudal arch stage. So the soybeans are just coming through the ground. Okay, so we're going to take this back in the lab and put it in those pots, and we're going to have weeds surrounded the soil, and the weeds, and so there's the soybean plant's going to come up just as you saw the Arabidopsis sitting in the middle of that ring of grasses. The soybean will come up in that ring, and what you start to see is a change. The plant is able to, just as it's emerging, somehow it's able to detect above ground, which is amazing because it begins to alter its growth pattern if there's green tissue above the ground. And we certainly know that far red signals can penetrate to a shallow depth in soil. But if you look here, so there's no weed. If you look at the weedy one and you look at the weed free, and you look at the root system, you can see under the weed free, there's more fibrous root development occurring. And this is at the, just as the soybeans are beginning to emerge at the hypocotyl stage coming through the ground, okay? So it's already altered its structure, even though the weeds, there's weeds nearby, but not directly on top. And we can also see that it impacts on the root to shoot ratio. That is, there's less root being put into those that have weeds growing above it. That is, it's coming up into a green cover crop and it's getting more shoots because it's already preparing itself to defend itself to survive. Okay, so the root to shoot ratio begins to change. And we can detect at that stage, it's already upregulated antioxidants, okay? And so this is an example of a group of proteins, enzymes that are antioxidants, that their regulation has been upregulated because the plant's preparing to have to deal with this singlet oxygen. All this is happening as it emerges above ground. So two things. One, it's amazing about the speed at which the physiology of the plant is changing as it's detecting signals, that it's able to detect changes in its growth pattern and it's triggering antioxidant production. And so the antioxidant productions, let's think of those as sponges that's going to go out and absorb that singlet oxygen issue, okay, and help the plant survive. So very early on. So you may not see the damage in yield, it may have a time to recover, but I think when you plant green, there is that danger that you may experience high levels of singlet oxygen in plants. So something to think about, something to talk to farmers about. When I'm asked about it, I repeat myself here, but to say, I really think when a plant emerges out of the ground, it should only see its sibling. If you have a cover crop, and cover crops are all good, but cover crops can become a weed if they're not managed properly, okay? The second mechanism that occurs, and keep in mind now, this is occurring at the same time. So one, keep in mind, so we got the ball rolling. We've got singlet oxygen happening. The plant's got to go turn around and go, oh my gosh, I got to handle that problem. Added to that problem becomes the suppression of photosynthesis. Not only suppression of photosynthesis, but we can also impact the production of chlorophyll. So you get downregulation of chlorophyll genes. And so here's an example here of a group of genes that are related to photosynthesis. And you can see these ones are downregulated. That's why they're below the 0 line. And they're at a twofold level. So they're significantly impacted by simply the detection of a weed species. So you're going to start to downregulate photosynthesis. And I'm sure I'm quite confident I don't need to tell you how significant that can be and what that's going to cause. And so here, for example, chlorophyll biosynthesis genes that get downregulated. And we think that it might be occurring in the early stages of the chlorophyll production system. So, and we can take that and we can measure this in the field. This is some work done by one of my grad students that kind of profiled. This green here represents the critical period. On the left, you have CO2 assimilation. On the right, you have photosynthetic efficiency. And if you just look at the line here, that if you haven't controlled weeds until outside of the critical period of weed control, in the field, we can detect a rapid decline in photosynthetic carbon dioxide efficiency and photosynthetic efficiency here, excuse me, CO2 assimilation here and efficiency at this level. Rapid declines in those measurements on a plant-by-plant basis. So we can detect it in terms of genetics, we can detect it in the field. And in this study, the presence of weeds altered the assimilation of carbon dioxide, it altered stomatoconductance, it impacted on water use efficiency and photosynthetic efficiency and electron transport and rubisco activity. All critical variables involved in photosynthesis. And keep in mind, what's always fascinating me about this, there's no hammer in this story. There's no herbicide here that's going to cause destruction that you're going to see. This is a light signal. This is reading of a light signal and that signal having such an impact on the physiology of the plant. So how's that relate to other field conditions in corn? Let's come back to a field condition in corn, okay? So I'm going to relate some studies we did in collaboration with the University of Colorado, because we did these studies here at Guelph, and Colorado repeated the same studies. So I'm going to share that work with you here just to show you how it impacts. Because if you're going to impact on photosynthesis, you're going to dramatically impact on carbohydrate accumulation, plant weight, yield, right? So I'm going to show you here. So these are individual plants that are measured. And on the left over here, we have a harvest index. And on the x-axis, we have total dry matter per plant. And here's the frequency. We measured these plants. We measured these plants individually. This is the frequency of weight of those individual plants. And these little bubbles above there, those little circles above represent the harvest index. Okay, so that's the economic part versus the biomass part. So if I buy a hybrid of corn and I'm going to pay $350 for a unit of corn, I'm hoping that I've got a really uniform stand of corn out there with a common harvest index and to maximize my yield. Well, number one, that's not the case when you start to sample these plants, even though you have paid very expensive money for these top hybrid lines. Anyway, let's draw on the first one here under harvest index here. And what you want to do is you want to have a tight cluster here of the harvest index bubbles up here. And I want you to watch this green line. Okay, so this is yield is 100%. This has got a pre-emergence herbicide on it. It's kept weed-free throughout the season. And you can see here, we're sitting around approximately 300 grams per plant on average, okay? If weeds are present up to the first leaf and then removed, look at what happens. Yeah, the slide keeps skipping on me. You start to see harvest index is showing there a little bit, but you start to see it's pretty close to its 99%. Turns out that it was 99% of the yield. But watch what happens if you wait to the third leaf tip. Look how harvest index becomes a lot more variable. It starts to shift to the left here, okay? And look what happens here. This green line starts to move down. It's not as close to 300. Look what happens at the 5-leaf tip. Now we're way down below, and you can see, so the average weight per plant has started to dramatically increase. And this is changing by the leaf number, okay? A leaf number in corn and under optimum growing conditions will come out every 3 to 4 days. So you're getting this, again, this shows the dramatic increase in speed at which a plant is capable of changing. Look at the spread here of weights. It's no longer uniform, it's starting to fall all over the place. Harvest index is starting, look, we've got starting to shift to the left. And if we haven't controlled weeds by the 10th leaf stage, we're down now to about 150 grams per plant. And the one on the right, if we didn't control our weeds at all, you can see here that harvest index has basically started to collapse. This is like Niagara Falls, right? Harvest index is rapidly collapsing. The plants are barren at this point, and the average yield is down from 100, below 100, from 300 to 100, okay? So carbohydrate accumulation, photosynthesis, all impacted dramatically when you start to play with photosynthesis. And the fact that we can do that just by having the presence of a weed species is mind-boggling, absolutely mind-boggling. These are just signals that changes everything. So we get a reduction in carbohydrates, we get increased variability in harvest index, weight and size of the plants decrease, and clearly we know that yield declines extremely rapidly. So very dramatic, very fast. So, you know, you can actually think about weed control. If you start to calculate, you can calculate yield loss. If you look at some of the graphs, you can calculate yield loss on a per minute basis. So if you start talking to farmers about, you know, they'll say, well, I might leave those weeds till Monday before I spray them. And you start discussing the impact of that you can calculate yield loss by the minute. That changes the way you start to think about how you manage a field of with weeds in it, for sure. Okay, and we believe our data says that's for real. That's for real. The other mechanism which we've recently discovered, is that you can actually increase the susceptibility of a plant to insect and disease invasion. Okay. So the presence of a weed, and I put this in Star Trek terms again, shields are down. The shields can no longer protect you from incoming. Okay, the shields are down. The plant has sacrificed, it appears anyway, that the plant sacrificed its ability to defend itself against insect and diseases because it has to grow and it has to control some of these other problems it's having. And so it starts to give up on vital mechanisms. It seems to have a mechanism, a way of adjusting itself to do that, which is fascinating that it would do that. Okay, but the shields now are down, all because it saw a weed species, right? And so to explain that, jasmonic acid is an important part of a plant, as a plant hormone that plays a key role in defense mechanisms against our insects and diseases and abiotic stresses. And there's a gene, in particular gene that we followed here, this ST2A, it's sulfin transferase, which is capable of reducing the concentration of bioactive jasmonic acid in the cell. There's a related gene I mentioned here. We don't know the mechanism by which that one tends to work. But here's what happens is if you upregulate that ST2A gene, and so here we are under low red biological system, you can see that that gene has been activated. It is highly activated. Interestingly, it's activated again, but not as much under the far red light itself, the artificial light, and we both get activation of ST2B as well. But the ST2A gene can, the upregulation of that will mean that it's reducing the hormone levels to jasmonic acid levels. And when it does that, it represses the bioactive GAA. And what we think is happening is that it's promoting the shade avoidance response that by lowering JA levels, that triggers, that helps. It's one of the mechanisms that contributes to the shade avoidance level. So it's going to grow, and it provides it a mechanism to help acclimate, that is to reduce the level of singlet oxygen. So it chooses to grow rather than die from insect or disease. So it sacrifices its ability to defend itself in order to control the singlet oxygen problem and to get growth going as a mechanism of survival. Very unique and very interesting that it's capable of doing that. The last discovery that we made is that we find that the presence of a weed species, the signal of far red from a weed species, can cause nitrate accumulation in the leaf and the roots. It affects assimilation, never ever been reported before. Okay, so this is the first report of this ability. So you change how the plant's handling its nitrate. Okay, and we didn't know about the mechanisms. We're just working on this manuscript now. So we looked at major enzymes in the nitrogen assimilation pathway. And we were looking to see, and we were thinking initially that it might be stored in the vacuoles. That's the first step. And maybe nitrate transporter genes were involved. So if we just call it transporter genes, like the ferry boats across the membranes, the ferry boats aren't running, and genes are starting to, or excuse me, nitrogen is starting to back up in the plant. So here's some of the data. So on the left here, we have nitrate accumulation. We're talking about leaf tissue here. Here's our control, which is a weed-free one. This is where we have the weeds present, and you can see that we get an increase in nitrate concentration in the weed species. Sorry. And under low far red light as well, for the artificial light, we get the same type of thing as well. This thing moves like crazy. So we see that, and we see the same response to weeds in the root system. So we're affecting nutrient transport here. We're showing you nitrate. It could possibly be other nutrients as well, but we're focused here on nitrogen. Okay, this has never been reported as an impact of competition before. either of these other mechanisms, but this one was just fascinating here. So we looked for transporters. That was our first approach. We thought, well, maybe transporter genes are impacted. And we did not find any distinguished genes that were expressed, okay, under biological or artificial red low light. So we eliminated that variable. But we started to look at enzyme activity. So here's the nitrate pathway. And we looked at these enzymes here: nitrate reductase, nitrite reductase, and glutamine synthetase, and FD and NADH in the GOGAT system, okay? And when we analyzed for those systems, we found that nitrate accumulated in the leaf. Nitrate reductase activity was equal, no change in that. Nitrate reductase activity, there was no change in that. There was no change in ammonium levels, and there was no change in glutamine synthetase levels. But FD-GOGAT, the enzyme in the GOGAT system, was downregulated, and that downregulation could result in accumulation in the root system. And so here we looked at these enzymes, and here we find that in leaf tissues we get downregulation. Here we get the litivirus decreased GOGAT activity in leaves, but did not decrease it in root tissue. So there's the NADH is part of the root tissue system, and so it did not have the same mechanism in the root tissue. So we did not suppress it there, but we did suppress it in the leaf tissue. So interesting. So we're still working on trying to figure out how all that comes together. So in conclusion here, I want to say that what I've tried to show you is that part of competition involves these physiological changes, that it is more than resource dependent, that you can increase cellular singlet oxygen, and that increase in singlet oxygen requires energy to handle it. There's an acclimation response, and that leaves a legacy impact too, because it takes a time for the plant to recover from that damage. You can reduce photosynthesis. You allow the plant, you get a reduction in defense mechanisms against insect and diseases, and you can cause nitrate accumulation and impact assimilation over time. All those four are happening at once in the plant, okay? These are not sequential. We don't know which occurs first, but we do know they're not sequential events. They are all occurring, and all of these will require energy. So energy expenditure has to keep, I think it comes down to energy at the end of the day, because the plant has to expend energy rather than in growth and development and maintaining a proper allometry. It is now trying to solve all of these problems at once. All of these headaches arise at once simply because there's the presence of the weed species. So what are the research implications of this work? One, it certainly provides novel and new mechanisms for understanding about how plants compete against one another, okay? And it opens up a whole world around what can be done, okay? So primarily, in terms of practical weed science, it provides the science behind, our recommendation: control weeds early, start clean, stay clean. Best recommendation you can ever have in weed science. And if there's a theory in weed science, that's a theory. Down the road, it may be possible if we understand these mechanisms to breed, more tolerant crops to weeds. If we could shift the yield loss curve ever so slightly to the right, rather than, the longer the weeds being present, you get that standard curve. If we could just shift that curve a little bit, that would have a huge impact on overall productivity. And then it begs the question in my mind, and I put this out there because I'm getting too old to follow it up, but I would sure would if I was a young man. If we can get that much damage in a plant from far red, surely there must be some light signals that would help a plant recover from stress. So I would certainly explore that possibility. And that would be so cool if you could apply light signals to plants to help them recover from stress, to help them recover a few days earlier from stress. So some future ideas for future weed science students. And the last thing that I want to leave you with is that the one thing that this has really impacted on me is how common we are in biology, that we all share the same biology, whether humans, animals, or plants. And here I use an animal example because one of the students on this project was an animal science major. And when an animal is stressed, and I would think that you could equate this to our human experience when you go to the hospital, or even when you go for a medical, they talk about the white coat syndrome, right? Every time I go, my blood pressure goes way high, and the doctor says, "Hey, at your age, you got trouble." And I go, "No, I don't. That's just your white coat." So we all know, I think we experience this type of thing when we get under stress, whether you're an animal or a human, and you experience things like oxidative stress, reduction in carbohydrates, enhanced antioxidant activity. You get increased risk of cell death, and it causes altered nutrient uptake on translocation. That is what stress does to an animal. It does that to the human body. So I've shown you today in a plant, it shows a reduction in carbohydrates, causes antioxidant activity, increases cell death, alters nutrient allocation, and importantly, causes oxidative stress. So if there's one thing you remember, well, two things I want you to remember from the top. There's more to competition than resource dependency. And secondly, we share one biology. And I think that's almost a spiritual moment for me. So with that, I want to thank you, the audience. I want to thank my people who allowed this research to be done. And I want to thank you for being here. Merci beaucoup. And I'm happy to take any questions you might have.
Thank you, Clarence. That was fantastic. I sense that there's some blown minds in the audience here, and I really appreciate all the Star Trek references. Yeah, so let's hear some thoughts. I see Elizabeth has her hand raised. Yeah, go ahead. Yep.
Hey, Clarence.
Hey, Elizabeth, how are you?
Doing well, you?
Good, nice to hear from you.
Yeah, same. So you talked about the downregulation of a lot of the photosynthesis and chlorophyll genes when the plants are really young. I'm wondering, is it that they're too young to have some of the mechanisms built out yet to handle the single oxygen, and that's why they're unable to recover with time? Because they just haven't had time to build that physiology and biochemistry pathways out sufficiently?
Good question. See, okay, so the problem with my research is it's a snapshot in time. Okay, this is all like corn up to the 4th leaf tip stage, or soybeans up to the 3rd trifoliate stage, that type of thing. So it's a snapshot in time. We don't have, I can't answer the question, how long does that recovery take? Is the plant capable of recovery? The one thing we do know, though, is that the crop, as weeds emerge later in the growth stage of a crop, it has a dramatically different impact on the yield loss associated with that crop. And that changes by the growth stage, by the leaf stage, okay? So again, I want to stress the importance of understanding the significance of time of emergence, the time weeds that emerge with the crop, that is at the seedling stage of the crop as well. They're highly competitive and they're causing some of these dramatic changes. But if those same seedlings at the same density emerge when the crop is, say, at the 4th collar or 5th collar level, that's a totally different yield response curve, right? So I think there's something to that. I think as the plants get bigger, get more mature, they're able to handle that interaction differently. How they do that, I don't know. So that's as far as I can take that, Elizabeth, I think.
And Clarence, I had turned off your video to save bandwidth earlier. We were having a little bit of stuttering with the bandwidth, but yeah, if you want to try again with turning on your video, that would be great. Are there other questions? Otherwise, I have a few, but yeah, feel free to unmute or write them in the chat. Yeah, so Clarence, I know there's some folks on here that are interested in planting green, and I've mentioned your work to before with folks like Matt Ryan, and some of his lab members are here. Who do a lot of work with planting green, and they haven't seen much impact of that early season far red light on the crop. Do you think that there's a level of just adaptability in overcoming this early adversity that maybe there's definitely an effect there, as you've shown, but can it be overcome in more real world situations?
Yeah, I think so. So that the norm would be for that singlet oxygen to be, the plant will try to compensate for it.
Mhm.
Okay. I think that if you plant green, so it all depends what you're doing when you're planting green. If you sprayed, say, glyphosate, 5 or 6 days before, and the plant is dying, that's one thing. Or if you've just crimped say the rye or whatever the cover crop is, and it's going to stay green longer, I think those variables might come to play. So these initial impacts, so the longer that it's green, I think the more significant the impact. But it's quite possible that the plant, that this level of damage gets compensated for early on, and the plant is able to recover. See, that's the point. I don't have a picture of that recovery in the plant, and that work needs to be done yet. So it is possible that there could be some compensation that allows the plant to overcome that damage, but and the initial damage may be not enough to have impacted on yield. It's possible, but I think you have to be aware that if you combine stresses, let's say you do that and you're into a dry spring, I think then you could have, you've intensified the stress levels, then I think you could have significantly more damage. So it may depend on a number of variables in terms of the environment as well.
Great, thank you. Yeah, any other questions or comments? I was also curious about the proximity. To some extent, there's always green around us. Whether it be a nearby forest or a weed, a few inches from the crop.
Right.
Is there an attenuation factor of the far red light going through the atmosphere?
Yeah, and weed species differ too, whether you're talking a monocot or a dicot, because it's about leaf area. It's about how much leaf area do you have to send a signal,
Mhm.
and how close that leaf area is to the plant. So yeah, the further that is away. So we're relatively close to the plant to get that signal. So the signal is fairly strong, and the red to far red ratio, you got to have it down low for sure. Yeah, the proximity and the weed species will have an impact on the signaling as well.
Great. Did that spark any questions in anyone here?
Hey, Clarence, this is Tony DiTomaso. Clarence,
I
I
thought
thought,
you were the dean. I thought you were an upper mucky muck at Cornell.
oh,
Still taking time for a seminar. Good for you, man.
yeah, I was going to say, Clarence, it strikes me just before I head out to give a lecture that, a lot of this work, we just don't include it in our teaching. We're still talking about, like you said, competition for nutrients and light, which, as you say, are important. I'm wondering, when are we going to actually be able to incorporate some of this in kind of our more standard textbooks and make sure that students are aware of this kind of new perspective. I know, some people are still challenging it, but I also think there's a lot there that I think could be really helpful for the discipline. So just want to get your thoughts on that.
Well, I think anything that's new like this, so this is all new, okay? And you have to think, you know, competition is such a powerful source. It shapes everything out there in our communities. So there are going to be, you know, we had a response to a paper we published. So there's a, you'll see if you look up what we've published lately, we had to write a letter back to a criticism of our work. So it's like a trial balloon goes up in the air and everybody comes at it with a pin, right? They're going to try to poke a hole in it. So really this type of work, because it's novel and different, we have to have other people testing to see if it's true, right? You know, you can test it on a pepper plant, or you test it on an oak tree and see, you know, if you see similar responses. So, and that's the way science progresses, right? So you're going to have to see, other labs will have to say, you know, we'll test this, and eventually it's a yay or nay thing. So does the research we've done here have legs, or does it die off because, you know, nobody agrees with us? So this will take time, and we'll have to see how some of the papers get referenced and quoted and used over time. But I certainly think in terms of teaching, you can put these ideas out there as new ideas that still have to be further tested in a broader spectrum. You know, we work here on corn, Arabidopsis, and soybeans. And so we have in there, you know, a dicot and a monocot type thing. So we see it common there. There have been some work published in Weed Science that have shown the same results. So it's a beginning. But certainly if you know, grab part of this and talk about this as possible new ideas, put a grad student on some of it, you know, that has to happen over time. Or it will die for sure if it doesn't. Yeah.
Great. No, thanks, Clarence. That's I totally agree with you.
Yeah.
Great.
Yeah. Thank you, Tony.
Okay, we're going to end the formal question and answer period, and have people feel free to stay and kind of informally do a meet and greet with Clarence if you're a big fan of this new work, or perhaps some of his older work that I know a lot of us have read, the community assembly theory applied to weeds, which has been fantastic. So yeah, so thank you all for attending.